All right, let's dive right into this explainer. Today, we're going on a bit of a high-stakes quest to uncover exactly how scientists test and measure the real power of cancer-fighting drugs. So whether you're a 10th grade biology student or, honestly, just someone fascinated by the incredible science of medicine, you are absolutely in the right place. We're going to step into the shoes of a laboratory detective together and solve one of the most important puzzles in all of medicine, figuring out if a potential cure actually works. I mean, it's a massive question, right? How do we actually know a cancer drug works? Well, before a newly discovered treatment ever comes close to reaching a hospital or a patient, we have to play medical detective right here in the lab. We have to set up a tightly controlled environment just to see if a drug defeats our cellular suspects. We can't just guess or eyeball it. We need hard, quantifiable data. Now, our entire investigation basically rests on two main ideas: cytotoxicity and cell viability. Cytotoxicity measures the toxic impact of a drug, specifically how well a chemotherapeutic agent destroys living target cells. On the flip side of that coin, cell viability tells us how many of those target cells actually survive the encounter. And since not all drugs work the exact same way, analyzing this dynamic balance is absolutely crucial for developing new, effective treatments. Here's our lab schedule for today's explainer. We'll start with hunting for cancer drugs, move to counting with a hemocytometer, prepping the cells, running the drug treatment protocol, measuring health with an MTS assay, and finally, finding the IC50 value. Okay, step 1, hunting for cancer drugs and identifying our targets. Take a look at this lineup of mammalian cell lines. Scientists use very specific lab-grown cancer cells depending on the actual disease they're trying to cure. For instance, we might use MCF7 for breast cancer or the famous HeLa cell line for cervical cancer. Those are cells originally taken way back in 1951 that are, incredibly, practically immortal. Also, notice how each cell type requires its own specific kind of biological food, which we call media, like RPMI or DMMI, just to keep them alive and happy for the test. Moving on to step 2, counting with a hemocytometer, our microscopic grid. Before we can test any of these specific cells, we have to know exactly how many we have in our sample. And that brings us to a brilliant piece of lab equipment that basically acts as our microscopic city map, the hemocytometer. When you look at it under a microscope, you'll see it's divided into 9 major squares, each exactly 1 millimeter by 1 millimeter. To start counting, we carefully add just 10 microliters, which, by the way, is roughly a fifth of a single drop of water, right between the counting chamber and a super thin piece of glass called a cover slip. Now, because the chamber height is locked in at exactly 0.1 millimeters, the volume of a single major square section is 0.1 cubic millimeters. If we convert that over to a liquid volume, it equals exactly 10 to the negative fourth power millimeters. Think of this specific math as our lab cheat code. It effortlessly translates the tiny number of cells we can see under the microscope into the millions that are actually floating around in our tube. So, by following a few straightforward math steps, we can calculate our exact cell concentration. We just count the cells in the 4 outer corners of the grid, divide by 4 to find the average per square, and then multiply by that magic number we just found, 10 to the 4th power. Our ultimate goal for this whole experiment is to hit a highly specific concentration, exactly 5 times 10 to the 4th cells per milliliter. All right, step 3, prepping the cancer cells, or the action protocol. We kick things off by gently washing the cells in their culture vessel with a buffer solution just to clean them up. Next, we add a really cool dissociation reagent called trypsin. See, cancer cells love to stick to the bottom of their flask, and trypsin breaks those bonds so they float freely. Once they're floating, we transfer them to a tube and spin them down in a centrifuge, a machine that spins ridiculously fast, at 200 times the force of gravity for 5 minutes. This forces all the cells into a tiny, solid clump right at the bottom, which we call a cell pellet. Once that centrifuge stops spinning, we check the liquid on top, known as the supernatant. If it's totally clear, that means all our cells are safely packed in the pellet. We then carefully remove that clear liquid and resuspend the pellet in 5 milliliters of fresh, warm growth medium. To make sure our cellular suspects are totally equally distributed and not clumped up, we gently pipette the liquid up and down at a 45-degree angle against the wall of the tube. Next up, we take 100 microliters of that perfectly mixed cell suspension and place it into individual interrogation rooms on a 96-well plate. You can picture this plate kind of like a tiny plastic ice cube tray with 96 little circular compartments. Once our cells are seated in their wells, we place the entire plate into an incubator set to 37 degrees Celsius, which is human body temperature, and we just let them rest and attach for 24 hours. Now for step 4, where the real action happens, the drug treatment protocol, interrogating the suspects. First thing we do is suck out the old media they were resting in. Then we expose the cells to varying concentrations of our experimental drug, starting from a massive high dose of 2,000 micrograms per milliliter, all the way down to a low dose of 50. We always do this in duplicate, just to verify our results. And remember, as rigorous scientists, it is absolutely vital that we include positive controls, negative controls, and blanks on our plate. This is our baseline reality check, right? It ensures our final data isn't just a fluke. Once the drugs are added in, they go right back into the incubator for another 24 hours. After that 24-hour treatment period is finally up, we've got to pull off a pretty delicate maneuver. We have to carefully suck out the drug liquid without accidentally scraping or disturbing the cells that are attached to the bottom of the well. And why are we so careful? Well, because the chemical composition of the drug itself could actually interfere with the super sensitive color readings we're about to do in the next step. Which brings us to step 5, measuring health with the MTS assay. Basically, our biological lie detector. To find out if the cancer cells actually survived the drug onslaught, we bring in that lie detector. Here's how it works. We add a specialized 100 microliter mixture containing the MTS compound into each well and let it incubate for 2 to 4 hours. The chemistry happening here is just fascinating. If the mammalian cells are still viable, meaning they are healthy and alive, their natural biology will reduce the MTS tetrazolium compound, transforming it into a beautifully bright colored formazan dye. We then slide the plate into a machine called an ELISA reader, which shines light through the liquid to measure exactly how intense that color is at a wavelength of 490 nanometers. The golden rule here is honestly so simple. More color equals more living cells. If a well is deeply colored, it means the cancer cells survived, so the drug really didn't work well at that concentration. But if the well remains relatively colorless, bingo, it means the drug successfully destroyed the target cells. This makes the MTS assay an incredibly elegant, visual, and highly quantifiable indicator of the drug's true cytotoxicity. And finally, step 6, finding the IC50 value, the grand finale. All that careful pipetting, the microscopic counting, the high-tech color reading, it all leads us right here. It's time to crunch the numbers. That final absorbance data we pulled from the ELISA reader lets us calculate something called the IC50 value. This is a single, incredibly powerful metric. It tells researchers exactly how effective that specific drug was at stopping those specific cancer cells. By comparing the different concentrations we tested, the IC50 pinpoints the exact drug concentration you need to reduce cell viability by 50%. Basically, it's the ultimate grade on the drug's report card. So as you master these essential lab techniques, from mapping out cells on the hemocytometer to reading colors with the MTS assay, and you start piecing together the complex puzzle of cytotoxicity, I want you to ask yourself a question. Could lowering this single number, the IC50 value, be the exact key to the medical breakthroughs of our future? Keep exploring, keep questioning, because honestly, the next great lab detective could be one of you. Thanks for joining me on this explainer.